It's one of the latest technology which you can see in this world. And in this area, a cutting edge research in the area of medicines and also electronics and other areas are happening. And what is now nanoparticle? Let me just introduce the nanoparticle. Actually, any substance or any particle, the one dimension of that is less than 100 nanometer, then we say that this is nanoparticle, and the people who are working in that area, we call them that these people are working in the area of the nanotechnology. Why nanoparticle or nanomaterial are so important? Okay, so here we are. The reason is that what I just discussed now, and the reason is given here. Let me just give you a brief account on the history of the nanotechnology. Well, nanomaterial are there since the beginning of life. You know that the water and proteins and these are actually nanoparticle and nanomaterial. So you can say that that the nanotechnology was there since the beginning of life on this earth. And in 2000 years ago, that the sulfide nanocrystal were used to just stain the hair. So that's a very, very old technology. But the people were not aware of that. As you heard in earlier lecture about the gold nanoparticle, so you know that the gold nanoparticle were used to just prepare portrait and some sculptures, and these were extremely stable over there. But the real beginning of nanotechnology started after the very important and famous lecture of Professor Richard Feynman, who just delivered a lecture that there is a plenty of room at the bottom. So he mentioned that actually if that one day we will see an era where nanomachine will enter into our body, they will remove the unwanted material from the body without the use of scissor and knife. And that is the area where I am dreaming for. But at that time he was criticized by his fellow colleagues. But nowadays, in next one decade, I can see this is a reality. And these are some of the development which took place. So the real development of the nanotechnology started after the development of scanning electron microscope and transmission electron microscope, because then the people were just seeing the nanomaterial and they were also manipulating the nanomaterial. So that's how the developments started. So what is now, like you see just an analogy that the human hair is 80,000 meter thick, and 1 nanometer is 1 billionth of a meter. So you can see that the scientists who are working an object of less than 100 nanometer in size, so that called that particular person is working in the area of the nanoparticle. So those people who are working in the area of the nanoparticle, they are dealing with very, very small particle. Why this smallness is important? There are several reasons. That one, when you reduce the size from the micro regime to the nano regime, you increase the surface area. Once you increase the surface area, you increase the reactivity and activity of that material. Similarly, if you transfer a microparticle to nanoparticle, what you will achieve from that? You change the physical properties. Like the gold material, like you are having gold jewelries, so they are insoluble in water. But if you prepare the same jewelry with the gold nanoparticle, this becomes solubilized. That will give you a clear solution. Similarly, there are a number of other materials which are non-conductor. I'm just using the example of gold, and it's also not working. As you see that the gold is inert in nature, but if you if you convert this into nano regime, this becomes superconductor. So that is the importance, that you can convert the micromaterial to nanomaterial, and you can exploit their application. You have heard about carbon nanotubes. So you with a very small thin carbon nanotube similar to the thread where we can see our clothes. So with that you can hang an elephant. So I can see an area in next I would say in a decade that all these buildings will not be having steels. There will be threads of the carbon nanotubes, and they would be more stronger than the steels and the iron which we are using. Similarly, a number of other examples are there in the market, like L'Oreal is using nanomaterial. Solfresh, these are actually the socks which are used for the smelly feet of the people. So these are already there in the market, and a number of nano coatings are also there. Anti-staining coat, those dresses, if everyone would like that the dress should not get dirty, so there are like nano coatings. If you coat them, so your dresses will not be dirty forever. They will not catch the dirt. Similarly, antibacterial paint. Mercedes is using a nano coatings. So these are scratchless coating. Like even if you just use a bullet, still there will be no scratch in this. So they have already introduced this in the market. Let me just give you a glimpses of research which is taking place in my research group. We are actually working in the area of using nanomaterial to enhance the bioavailability of the poor water soluble drug. We have also used a number of our nanocarrier to reverse the resistance of the bacteria. Similarly, we have used them for the targeted delivery, and we have also enhanced the therapeutic efficacy. So these are some of the nanomaterial which have been developed in my research group. We have published them in these articles, and those since the time is very short. Similarly, we are from my research group, we have also written books in the nano carriers, and these are available online. These are some of the nanoparticles developed in my group, which we have been used for the treatment of the diseases, and also to increase the bioavailability of the various water insoluble drugs, and also for the targeted delivery. These nanoparticles have been published internationally in different journal. We have also written a book for the elsewhere on this. This is another book which have been contributed from my research group. Some of the example, we were able to develop oral formulation of the ciprofloxacin with the help of the nanomaterial which were developed in my group. The oral formulation of the ciprofloxacin is not available. It's available only in injectable form.
Can you please wrap up?
These are some of the antibiotics which and we were also able to just reverse the resistance of a number of bacteria with the help of the nanoparticle. The results are published in the reference for those. We have also just reported in this book which were contributed from my research group. This is another book which I have contributed, and let me thank actually my students and collaborators and funding agency and all for the contribution, and thank you very much for listening me.
Thank you so much, Doctor.
Thank you so much. This is on now. Thank you. I'd next like to invite Dr. Washdev Amir. He has worked in the Dr. Abdul Qadir Khan Institution of Behavioral Sciences, Dow University of Health Sciences. He is an expert psychiatrist with over 13 years of experience in his field. Today, he's going to be presenting. Okay.
Thank you. Oh, yes, I have. Oh, thank you. So, can you give me a pointer? Thank you so much. So, in 10 minutes, I am here with a question and concept. Substance use disorder, is it a brain disorder? As I said, I have only 10 minutes, so it's a big concept actually. So I can talk about only a little bit about it, of course not about the neurochemicals and so many things, and I need 3 to 4 hours to solve this query. Okay, what's the concept in community? It's a deficiency of character. These people just need tuning. It has to do nothing with disorder. If it's not with the disorder, if I talk about the ICD, International Classification of Disease, 10 and now 11 is in the market. So International Classification of Disease, all the disorders, they are defined in this ICD 10 and ICD 11. If substance use is a character deficiency, why this is in the substance use in this book? So basically, we have to solve and we have to see this. Is it a disorder? So if I talk about F is designed for psychiatric disorder in ICD-10, and 10219 is for substance use. 10 is for alcohol, 11 for opiate, 12 is for cannabis. All the substances are classified in this chart. For example, we have a local substance, pan, chalia, gutka. We can say that they are in the tobacco in F17. Powder, it's a mixture of opiate, it comes in the F11. Okay, so this is all about epidemiology. If you see in 2013, 6.7 million people were using substance in our country, and in 2018, 35.6 million. So see how the prevalence has raised in the last 7 to 8 years. It's more in the young people, and the cannabis has more prevalence as compared to the other substance in our society. So here is the classification of substance. So there are few CNS depressant, other are the psychostimulant, and few are the mixed. If I give you example in the community, you must have seen people on the pedestrian bridges, they are psychomotorily retarded and totally down. If you talk to them, they will talk to you, otherwise they are like Vex. So what they are actually using? They are actually using opiate. It's a psychodepressant, it's a CNS depressant. On contrary, if you come to the bus drivers, they hawk throughout the day, sadar, this, that, and kabhi bhi wo aap dekhenge thakenge nahi. And they will find a time to fight with their bus drivers. They are so high. If we talk about 20 to 30 minutes here and we need 1 hour break, why they are high? Due to this cannabis, because it has the psychostimulant effect. Similarly with the meth and ecstasy. And if I come to the other classification, all the substances have remembered psychological dependency, all the substance. That's why they are called psychoactive substance. Whereas few have only physical dependency, opiate, benzos, and cannabis, meth, they don't have physical dependency. The substance which have both dependence, they are more difficult to quit and complicated to quit as compared to those have only psychological dependency. I said that benzos have both dependency, physical and psychological, whereas cannabis has only psychological dependency. Do I mean that it's difficult to quit benzos, it's complicated to quit benzos? You must have heard the name, this lexotanil, alprazolam, and so on, as compared to the weed or anything else? Yes, it's difficult to quit and it's complicated to quit. So these are the local names. Every body function is a center in your brain, every body function. So if I come, substance use is associated with the reward. आप जब भी कोई substance use का बंदा substance लेता है, whatever you do in your life, अच्छा, यहाँ पे एक brain में center है nucleus accumbens, this is associated with the reward system. So anything you do in your life, it has जो भी dopamine release. You guys are here, dopamine. You get, you are going for sports, dopamine is released in your brain. You get first position in your exams, dopamine is released. Here, volunteers are here. When they come for a photo session here, your dopamine are released. Yes, we are doing something. So whatever task you do in your life, जहाँ पे reward होता है, there is a release of dopamine in your brain. Similarly is with this food, water, sex and nutrition. It acts like lock and key system. Every reward has its lock. So it, every your activity is key and it has its own lock. Now the misery is that the substance has master key. They don't have key, it has a master key. It has tendency to open all the locks. So actually, the reward from substance is more as compared to your other rewards. Your first position, your hair, and means you are going for games and so many other things. So when the person is using substance, so there is a myth कि जी पहले तो दोस्तों ने दे दिया, फिर ये क्यों उसने इस्तेमाल किया, या आगे क्यों इस्तेमाल करता गया. Actually, ये dopamine की वजह से, ये reward की वजह से. Now the 4 खाने are gone. Means your first position gone, your other activities are gone, and by time, substance. There is no enjoyment in other activities. If the more the dopamine released, more the brain will accept this. So the dopamine with substance is released more as compared to all the activities. So you must have seen the people in your community with the time. The person with substance use, ना वो games को enjoy करेंगे, ना अपने study को, ना वो अपने family members के साथ बैठेंगे, nothing. That's because of dopamine. So to counteract this, means we psychiatrists, without the help of psychologists, community members, we are helpless in this regard. So we have to counterpart this dopamine, this high dopamine. So you actually need a teamwork in the term of psychiatrist, general physician, your psychologist, your occupational therapist, students, rehabilitators, most important family members and the community members. Okay, many times there is a question, हमारे पास family members आते हैं कि जी हमें इसको सऊदीया भेजना है, किसी और country भेजना है, हम कैसे जो determine करें कि इनको substance use तो नहीं कर रहे. So there is a always query. So there is a test called as the urine for toxicology or urine for drug abuse panel, जो different laboratories में होता है. In Dow University we have urine for drug abuse panel, जिसकी cost only जो है 1000 to 1200 rupees पड़ती है, and we screen about more than 10 substance. So what's the goal? The goal of addiction treatment is to help the individual not only to stop or reduce the use of drugs, but also to make them productive functioning in their family, at their work, and their society. So for that, you always need motivation to determine कि आखिर अगर इनसे substance छुड़ाना है, तो इनके goals क्या है. अगर मैं ऐसे ही अगर किसी को admit करूं without determining goal, less likely है. And remember, relapse rate in the West is almost 85%. So जहाँ पे पूरी intervention होती है, psychological and rehabilitation, what you think about here? It's almost more than 95%. Why? Because you don't determine goals. So here is we have few papers measuring treatment readiness among patients with opioid use. Brigadier Saab is the part of that paper. So we have done this study in Dow University of Health Sciences. Another paper, survey to determine motivating factors. You can go through these papers. These are free. So what we have seen in 10 minutes, although I will again say that to prove that I need 2 to 3 hour workshop session for that. It's not a character flaw or personality disorder, it's a health problem. And you need to motivate the people in your surrounding who are using substance. So if you go after this your session, when you will go to the Mosmiat, you will find outside Dow Lab, and beside Dow Lab, a beautiful parliamentary building. It's a Dr. A.Q. Khan Institute of Behavioral Sciences, where we work. It's the department of psychiatry, DIMC. Brigadier Saab, the head of that. और यहाँ पे एक जो है ये ब्लैक गाड़ी भी खड़ी हुई है, वो भी वहीं पे खड़ी होगी. So say no to drugs. Thank you so much. I guess I have done in 10 minutes.
Highly appreciated. Thank you.
Thank you.
Thank you so much for that super interesting talk, Professor. Next, we're going to ask you to direct your attention to the screen for Professor Wagan. It's going to take a second to connect, but he is going to be talking about the story of coffee and Parkinson's disease.
You can start, Dr. Wagan. Can you hear us? Dr. Wagan, can you hear us? Dr. Wagan? Assalamu alaikum, Dr. Wagan. Can you hear us? Can you text or chat with him and say that he is online and now he can start his talk?
Hello, Professor Wagan, can you hear us?
You are mute, Dr. Wagan. We cannot hear you.
You're muted. We can't hear you yet.
Still
We can't
muted.
hear you yet, I'm sorry.
We can't hear you. No, we still cannot hear you, Wagan.
I'm sorry, we still cannot hear you. Hello.
Let's move to the next one.
Unfortunately, there's some tech issues going on, so we're going to go ahead and move on with our next speaker, who is Dr. Saima Mahmood and Dr. Shabana Usman Simji. So sorry if I pronounced that wrong. Dr. Saima is an assistant professor at DUHS. She is an Eremia Scholar from the Department of Pharmacology Science and the University of Milan, Italy. Presenting along her side is Dr. Shabana Usman, who is a professor at the International Center of Chemical and Biological Sciences at the University of Karachi.
Okay.
Please start.
Okay, Assalamu alaikum guys. Thank you very much for all the organizers, especially the organizers from Pasman, Khan University Hospital, Dow University Hospital, Dow International Medical College, Biochemistry Department. I welcome you all again and be patient with me. I will try to concise what I want to talk to you. It's basically, I want to give you an idea that using the basic tools, how we can go for the newer drug discovery. We don't need major rocket sciences. By just gaining the knowledge of the basis, by utilizing the available resources, we can do a lot. And many of the researchers, they say so. So I just want to summarize it here for you people. Many of you may be working previously or early on those apparatuses by using those animal models, okay? But I would try to go ahead. This is a short description, that is neuropharmacology screening tools to discover newer therapeutic agents. Okay, it's basically the outline of the description, that is starting from the background of the diseases. I will try to summarize selected 5 diseases, their the drugs, available drugs, their mechanisms of actions, and in comparison to that, how we can use multiple toxins or drugs to induce the diseases in animals, and evaluate or assess some criteria, and that may lead to discovery of newer agents. So starting from the definition of pharmacology, most of you be knowing that, but it's basically explore the various aspects of drug discovery, development, and its preclinical drug safety. Hence, neuropharmacology deals with the effects of drugs on central nervous system diseases. Regarding the screening, multiple screening tools would be utilized against a chosen target for a particular disease, but this may be highlighted as high throughput screening in vitro. But again, we can utilize this as a basic initial tool and ultimately go for preclinical studies. Historically, I just want to identify this man, Frederick Serturner. He isolated in 1805 the morphine from opium plant. Dr. Vasudev was discussing about the substance use, and morphine is can be highlighted as one of those. It can be misused. He was the person who isolated it from opium plant first time, and he used it by himself and studied the effects. Hence, it was named as Morpheus, on the name of the Greek god of sleep. This is a small summary of how the drug discovery and development process goes on, starting from the early drug discovery. The next one is the preclinical studies, then the clinical development, starting from phase 1 to the phase 4, then the filing of the application for FDA review, and then the post-marketing surveillance. I would be talking about the preclinical studies here, in vivo models. Okay, for screening agents, as I have summarized you, we would be identifying what are the in vivo models for those 5 selected diseases, and for the better understanding of various therapeutic strategies and how we can discover your agents from those. Okay. This picture gives you a summary of mechanisms of actions of anti-Parkinson's drugs available in the market. At least 4 can be identified here. That is direct action on the dopamine receptors, or by inhibition of catechol O methyltransferase, or by inhibition of monoamine oxidase B, or directly that is the chance of levodopa. It crosses the blood-brain barrier, reaches the brain, and ultimately divided into dopamine. Further, mechanisms of action of anti-epileptic agents. Here is a summary. The mostly 3 major mechanisms of actions are there. That is prolongation of sodium channels inactivation, facilitation of GABA-mediated chloride channels opening, and the inhibition of T-type calcium currents. By acting on these channels, anti-epileptic drugs, they produce their actions. So, but still, by having said that, although we do have many of the available medications, but we don't have exact or full cure of the diseases. About 33% of the patients are resistant towards these available medications. So we need better and newer drugs. For that purpose, here there is a summary that we can utilize the screening tools for Parkinson's disease, few of the drugs and toxins. By utilizing these toxins, by using various routes of administrations, either orally, intraperitoneally, or subcutaneously in specific doses for specific time period, we can assess them by few apparatuses, rotorod, pole test or catalepsy bar test, etc. And we can have this opinion that either the animals are, they have become fully Parkinsonian, or they have showed few of the symptoms of the Parkinson's, so that they can be compared against the standard medications available. Similarly, for epilepsy, we can utilize many chemoconvulsants. We can utilize maximal electroconvulsive shock therapy also, but few of the toxins are enlightened here, that is strychnine, picrotoxin, pilocarpine, etc. And for the evaluation of this, we can use Racine scoring system along with the observation in the electroconvulsive methods. Sorry. Okay. The tools for assessment for epilepsy, that is the Racine scoring system. It started, the scientist Racine introduced it in the 1977, starting from 0 to up till 5. 0 with no effect, 1 with a single wave of myoclonic jerk, or the 2 mild twitches, 3 multiple myoclonic jerks, and the 4 leaning down towards one side, and up till the 5 or 4.5 full tonic-clonic seizures would be developed. Although this is the observational studies. Here you can see that this is the fully tonic-clonic animal by maximal electroshock seizures. In that case, we can put electrodes on the ears of the animal, and we can give for a few seconds 0.2 milliamperes electric current, and ultimately the animal would become epileptic. And it can be compared or reversed by giving standard anti-epileptic drugs or anti-Parkinson's agents.
Dr. Saima, you need to wrap up quickly.
Yes. These are the few of the apparatus we can utilize to identify either the animals are Parkinsonian and how they would be compared with the group of the animals which have been treated with the standard anti-Parkinson drugs, and the other third group would be of the test material. So we can have a lot from these basics utilization, we can have scientific data on that. Similarly, anxiolytic agents, their mechanisms, as you all know, GABAergic mechanisms are there and serotonergic mechanisms are there. And regarding antidepressant actions, tricyclic antidepressants and SSRIs, selective serotonin reuptake inhibitions are the mechanisms. But we can evaluate it by using these simple tools, that is open field or elevated plus maze. By utilizing these apparatuses, we can have anxiolytic activities in newer agents, and similarly by using the forced swimming test or tail suspension test, we can have an idea of how desperate the animal is and how the newer agent would be improving that one. This is the last one. Many apparatuses are there, but for Alzheimer's disease, you can clearly see that in the first trial, the animal would be wandering here and there, and it would take longer time period to reach that platform. And ultimately on 8th trial, it would be directly going to the targeted platform. So this is
Please
the summary of our work. You can have on internet by googling my name, or you can have all this information. This is the summary of our lab work at Dow College of Pharmacy. We are working on these areas with all our faculty members. Few glimpses of our research facilities there at Dow College of Pharmacy. And I would like to acknowledge my mentors at undergraduate level and at my postgraduate level, Madam Tasneem Mariam Sajid from Bakai Medical University and University of Karachi. Dr. Shazia Qasim Jamshed from Bakai Medical University. Right now she is in Malaysia. Professor Dr. Shahid Rashid, retired from University of Karachi and Bakai Medical University. Late Professor Dr. Sayyid Naimul Hassan Naqvi Sahab. And right now my research supervisor, from where I have learnt a lot, that is Professor Dr. Mohsin Raza from ICCBS, University of Karachi. Then the late Professor Dr. Shahida Parveen, and currently my supervisor, Professor Dr. Shabana Usman Simji. Thank you very much for your patient listening and giving me the time.
Thank you so much.
Thank you so much once again. Do we want to give Dr. Wagan another chance before we go ahead with the next one, or should we head straight in? Okay, can we, if Dr. Wagan's online, can we please try to get him back on? Dr. Wagan, can you hear us?
Slides and
We can hear you.
I can hear you perfectly.
Finally,
perfect.
welcome.
Perfect. Yes, my hate for Microsoft Teams increased immensely today. So thank you so much for inviting me. I'm sorry for the earlier troubles. My name is Imran. Happy to be sharing the same platform as Dr. Shan Siddiqui and Dr. Thema. It's a huge honor, huge fan of the work. So my title is a bit clickbaity. It's basically not just the story of coffee and Parkinson's, but the story of what my lab has been doing for almost the past decade. So here are my disclosures. So just to jog your memory a bit, what is Parkinson's disease? So basically there is degeneration of these neurons sort of right here in substantia nigra pars compacta, right here, and they give dopamine to striatum. When there is no dopamine, of course, that leads to the imbalance of the striatal output, which ultimately leads to the Parkinsonian symptoms. You may have seen this slide like a million times in your medical school, but let's go through it again. There's this D1 receptor, but what we are more interested in are these indirect pathway or the D2 receptors. And specifically along with these D2 receptors, there are these A2A receptors. If you've been paying some attention to Dr. Adhainan's course, Essentials of Neuroscience, you might remember, I think it was lecture number 6, where he talked about purinergic signaling. So, here are the A2A receptors. So what do these A2A receptors do, and why are we interested in them? These A2A receptors, they form sort of this antagonistic heterodimer with D2 receptors. So basically what that plain and simple terms means is that whatever D2 receptor does, the A2A receptor tries to sort of antagonize it and do exactly the opposite thing. So when we are trying to give dopamine agonists, we are sort of manipulating the D2 receptor and the output of the D2 receptors. But one could also assume that if we somehow manipulate A2A receptors or antagonize A2A receptors, that could potentially have the same effect, which ultimately leads back to 2012 when there was a study that suggested that there was that Parkinson's disease symptoms, they kind of improve after patients are given coffee capsules, which led my supervisor, Professor Laura Ayers, to think that we should probably investigate this with PET, why is it happening in the first place, right? With PET, what we try to do basically is this like a very busy slide, and apologize for that. But we had 6 healthy controls. We had 10 early stage PD patients and 10 moderate stage PD patients. They underwent this PET scan to target the specific A2A receptors so that we can see what their availability is in the brain in Parkinson's disease patients as compared to the healthy controls, right? What we found, there was some evidence already when we decided to start our study. Ramla Khan Singh and colleagues, they found that there was this increase in the late stage Parkinson's disease patients, the dyskinetic stage, but there was no evidence in the early and the moderate stage. We found out that there was this decrease in coded A2A receptor availability in the earlier stage. But when we sort of looked at the moderate stage, we found that there was this, I'm sure you can appreciate, I mean, it's pretty hard to see, but there was this increase in A2A receptors expression in the pallidum. I shouldn't say expression, I should say availability, because it's sort of like an indirect measure with PET. So there was this increase in the pallidum A2A receptor. What does that mean? So if A2A receptor is like a brake, so what we can assume is that when the patient was taking coffee, and coffee is an antagonist of this A2A receptor, that brake was being released, and probably that's why the patient's symptoms were getting relieved in the short term. But my supervisor was kind of interested in a more sort of interesting question of why coffee is neuroprotective. So coffee drinkers are said to be protected from not just Parkinson's but also Alzheimer's, and we were kind of interested in that question. The first clue that came was not from Parkinson's but from MS. My supervisor, Dr. Aero Rizinen, he investigated in secondary progressive multiple sclerosis A2A receptor expression, and he found that in white matter, I'm sure you can see here, in white matter there was this increase in A2A receptor expression as compared to the controls. Now, you must be thinking that, well, multiple sclerosis is a neuroinflammatory disease, and we're talking about Parkinson's, which is neurodegeneration, but neurodegenerative disease. But you'd be wrong. Parkinson's is also a neuroinflammatory disease. In 2006, there was this paper. One of the earliest findings with PET that they found out that there was this microglial activation in the frontal areas and somewhat in the temporal and the parietal areas as well, right? And this basically led my supervisor to Yale. She was doing her sabbatical there, and she was not convinced that like coffee, that we still don't know how coffee is somehow protecting against Parkinson's. And she had this idea, she being a neuroimmunologist herself, had this idea that maybe A2 receptors are expressed on microglia or perhaps on astrocytes, and maybe that's how it is conferring that neuroprotective effect. Indeed, with Dr. David Pitt at Yale, we found out that A2A receptors are indeed present on something called reactive astrocytes. So you have like this A1 astrocytes and A2A astrocytes, A1 being the protective ones and A2 being the sort of the destructive ones who release these neuroinflammatory mediators like IL-1 and as-CQ3 and so on and so forth. And you can appreciate that with GFAP, which is a marker of reactive astrocytes, are expressed on the same astrocytes on the same astrocytes in cortex as well as the perilesional white matter, which ultimately led my supervisor to ask me whether a similar thing is happening in Parkinson's. So we already had the data, and we decided to sort of look into the white matter in Parkinson's. And indeed, in the same areas where David and colleagues found microglial activation, specifically the frontal and parietal areas, we found that there was this increase in A2A receptor expression in both the early PD patients and the moderate PD patients. We also found that there was this association between increased frontal white matter availability and lower frontal low volume. So suggesting that maybe A2A receptors have something to do with it. But we were still not sure whether whether they're on astrocytes in Parkinson's, whether on some whether they're on endothelial cells. We don't have a clue. So what we do is we do this immunohistochemistry in the cortex and white matter, and we found that indeed.
Doctor, I'm so sorry to disturb you. You have 2 minutes left.
All right. So indeed, A2A receptors are present in the cortex as well as in the white matter. as I'm sure you can appreciate. So what is the moral of the story? Well, we like to believe that indeed A2A receptors are somehow, or A2A laden reactive astrocytes are somehow driving the neurodegenerative process in Parkinson's. But as Daniel Kahneman in his book Think and Grow Rich, not Think and Grow Rich, but like Thinking Fast and Slow, he says that you have to be aware of your biases. So there could be a very simple explanation. Maybe what coffee is doing is in relieving the constipation. We know that alpha-synuclein actually goes from your gut and towards the brain, and maybe coffee is doing that, and it is the ultimate explanation is not as complex as we or I may be suggesting with Moran data. With that caveat, I have to put this here. One of the people that I really admire, Professor Alberto Espe, he says that Neil deGrasse Tyson famously quipped that the universe is under no obligation to make sense to us. The same goes for biology. So yes,
Imran, can you please wrap up?
although the almost a decade-long work of ours suggests that this might be the case, but we have to be very skeptical of our own work. And yeah, so that's pretty
Please
much
wrap up,
it for
Imran.
me. Thank you so
Wabin,
much.
I am so sorry to disturb,
And,
but your time is up.
Yeah, there you go.
Thank
Thank
you.
you so
Thank
much,
you
and
so
I'm happy
much.
to
I'm
take
so
any
sorry
questions
once again,
later
but
on.
thank you so much for your speech. I'd like to now introduce Dr. Sonia Siddiqui to come up on stage. Not only is she the overseer for all of this, she is also a PhD who went to Ruhr University Bochum and is currently an associate professor at Dell University of Health Sciences. Today, she will be presenting her presentation, Extracellular Matrix Protein, Potent Targets to Develop Therapeutic Drugs. Please come up on stage.
Okay. So today we're going to discuss a little bit about extracellular matrix proteins and their role as to target, to develop therapeutic drugs. So normally what will happen, extracellular matrix proteins, you all know, you are all medical doctors here and medical students, we usually talked about extracellular matrix normally in every class. So they are actually the proteins that are present around the cell, and these are the molecules which are very important because these molecules helps in the neurite outgrowth, in neurite guidance and proliferation of different cells. Important thing is that when there is a development of nervous system, these extracellular matrix proteins, they help neurite to grow and to guide. But once the neurons start to degenerate, or if you have a brain injury, and when the brain development is stopped, these extracellular matrix proteins, they become more non-permissive than rather become or remain permissive as they were during the development. When there is a brain injury, these extra matrix proteins, they surround the gliotic scar and surrounds the nerve injury or the injury site and becomes the main culprit of the gliotic scar. Now the gliotic scar contains several type of different matrix proteins such as tenascin C, tenascin C, laminin, and fibronectin, and many other non-permissive and permissive molecules. So we usually target and molecules and biomolecules and therapeutic agents that usually target to reduce the expression of these extracellular matrix in the nervous system when there is an injury is act as an anti-inflammatory and helps in the development of regenerative medicines. So in search of different potent compounds that have ability to increase the neurite outgrowth, because normally after the injury neurons remain alive, but they do not make synaptic connections because of the presence of non-permissive extracellular matrix proteins that surrounds them. So they did not overgrow and they do not make synaptic connections with other neurons. So we have my lab usually targets metacatic acid, gallic and vanillic acid, and we also target piperine, and we identify their effect in different scenarios that they have. If they have neurite outgrowth effect, they have anti-inflammatory, and they have also neurite and helps in the remyelination of the neurons. Okay, so we also target, one of the molecules that we target is protocatechoic acid. It is presently present in, it's basically flavonoid. It is present in strawberries, it is present in honeys, in raspberry, and in white grape. So it means that it's present widely in your food substances that you usually take. And its structure is resembles to the antioxidant molecule. So we wanted to see that whether this molecule have ability to increase neurite outgrowth and have ability to reduce the expression of non-permissive molecules and pro-inflammatory markers after in the model of neurodegeneration. So we have created an ex vivo model of neurodegeneration by treating the cells with 0.003% lysolecithin. And we co-cultured and we do it by using neuroglya co-culture. So where we culture the neurons that is being shown in the green, and we also, obviously they are myelinated, so we have used oligodendrocytes that helps in the myelination, and we have stained them with the green one is the neurite and the red one is the MPP positive oligodendrocyte. So we have neurons, we have oligodendrocyte, and we have astrocytes in the culture. So this is the parameter. So what we do, we actually coat the cells, and then after 1 day we add the hippocampal neurons. We use hippocampal neurons, and then after day 3 we actually we provide them the lysolecithin, and then we divide into 3 groups. Then after day 12 we add, after 3 days we add our compounds, and we see whether they have an effect on the neurite outgrowth and remyelination of the neurons. So first what we did, we actually see different concentration of protocatechoic acid grown in different concentration of neurons grown on protocatechoic acid in different concentrations. So all the concentrations that we have tested, we identified that protocatechoic acid has ability to increase neurite outgrowth. Then we, and both the mean length of 10 longest neurites as well as the sum of longest neurite, they all are activated, and they are then we use current clamp modes to see, because when the myelination or when the demyelination takes place, so it means that it also affects the sustained repeated firing. Okay? It also takes place. So we actually record the through current clamp, we record the sustained repeated firing. So what we did, we actually expose, or we give continuous supply of 50 picoampere of the current for 400 milliseconds to the neurons, and when the neurons burst and activated and burst the series of sustained repetitive firing. So in current clamp mode, we hold the current that is passing through single line channels, and we see the fluctuation in the resting membrane potential, which is usually expected to change when you have demyelination. So we actually, okay, now this is the control, this is neurofilament. If you see the co-expression, in the control we see the co-expression of neurite with the myelin. It is abundantly present, myelin expression of myelin on the neurites are there. But when you expose it we treat it with lysolecithin, the demyelination takes place as well as the neurite also decreases. When you add protocatechoic acid in the in vitro model of neurodemyelination, you see the pictures, the co-expression of myelin as well as the neurofilament takes place. So in both the concentration of protocatechoic acid, it shows that it increases the neurite outgrowth as well as it also helps in the remyelination. Then we usually use, because after the addition of lysolecithin, the clear fibrillary acetic protein, or the astrocytes become reactive astrocytes, right? And due to these all act and start to express more and more reactive and non-permissive molecules, and becomes which hinders the neuron regeneration. So when we add protocatechoic acid, we see continuously the reduced expression of GFAP from the lysolecithin treated cells. So it means that these are the molecules that help in the reduction of these expression of these proteins. Chondroitin sulfate proteoglycan, we all talked about chondroitin sulfate proteoglycan. They are the most non-permissive molecules present in the gliotic scar. So if you see after the treatment of cells with the lysolecithin, the expression of chondroitin sulfate proteoglycan as well as the laminin also increases. However, laminin is more permissive, but after a while, laminin concentration remains the same, but the concentration of chondroitin sulfate proteoglycan, it increases day by day, which also makes the whole environment non-permissive, and the neurons cannot overgrow from this proteins. And when we add proteocassic acid, the expression of both laminin as well as chondroitin sulfate proteoglycan, they are decreased and remains with the control situation. Tennyson C, which is the most important and dual hexabranchian extracellular matrix, also shows the same, that the reduced expression in the presence of proteocatechoic acid, where if you compare it from the LPC treated cells. Then we also said that whether the signal transduction pathway, how the proteocatechoic acid act. So we usually use a protein expression that is being done by the STS page electrophoresis, and we identified that the protein kinase C, MAP kinases, and P38. So they are all, they usually have a decreased increased level when you treated the cells with the LPC, but when you added proteocatechoic acid, the expression decreases. So it means this proteocatechoic acid, they start to act by inhibiting through protein kinase C alpha, P38, and MAP kinases, mitogen activated protein kinases.
Dr. Sonia, can you
I still have 36 seconds. Thank you.
Yeah.
I have this stuff on. Wow. Okay, we also see the expression of pro-inflammatory marker, that is NF-kappa B, and we have COX-2, cyclooxygenase 2, which only increases when there is an inflammation. And both, after the addition of treatment of cells with these, with the proteocatechoic acid, the expression decreases if you compare it from the LPC treated cultures. Okay, this is the last slide. When we recorded the current, we have seen that the number of spikes and the latency to initiate the first action potential is also decreased. Okay? पहले ये था कि when we add lysolecithin, obviously the action potential initiation is increased, means बहुत देर बाद जाके action potential generate हुआ था. When we add proteocatechoic acid, action potential जो वो delay था वो कम हो गया. Okay? So this is also nice, and the number of spikes are also is increased if you compare it from the lysolecithin treated cells. Okay, the rest of the parameter of the action potential, they remain the same, means the action potential, resting membrane potential, and all. So I would like to thank my students who work day and night with me, and Dow University of Health Sciences, and the financial support from Higher Education Commission of Pakistan. So I
thank you so much.
Thank you so much for your presentation, Dr. Sonia. Our next speaker is going to be Dr. Thaniyat Faraz Ahmed. Dr. Thaniyat Faraz Ahmed is a lecturer of biochemistry at Dow University of Health Sciences and is a member of an international society to advance Alzheimer's research and treatment. Her research interests are in unraveling biochemical mechanisms and underlying pathogeneses of neurodegenerative diseases and using it to identify treatment strategies. For her work, she has been invited by the Alzheimer's Association at their annual conference in 2022 to deliver a presentation. Today, she is going to be speaking to us about plasma phosphorylated tau and neurofilament light hold promise as potential biomarker for Alzheimer's disease. So without further ado, please give her a warm welcome.
Assalamu alaikum. Thank you for this opportunity. Okay. Presentation. under the able guidance of Professor Dr. Fawzia Imtiaz, Dr. Uzma Zaman, and Dr. Naila Shahbaz, we investigated the plasma concentrations of A beta 42, phosphorylated tau, and neurofilament light as potential biomarkers for Alzheimer's disease. Alzheimer's disease is characterized by an insidious onset of cognitive decline, amnesia, and gradual loss of executive function, with the hallmarks of the disease being the extraneuronal deposition of amyloid beta plaques and intraneuronal deposition of neurofibrillary tangles of phosphorylated tau protein. The diagnostic criteria for Alzheimer's disease and the definition of Alzheimer's disease has evolved over time. The first diagnostic criteria for the disease was proposed by NINCDS, Alzheimer's Disease and Related Disorders Association, in 1984, and in this criteria, the clinical symptoms were supposed to always coexist with the underlying disease pathology. No use of biomarkers was proposed at that time, and the diagnosis could only be confirmed histopathologically. This criteria was revised in 2011 by National Institute of Aging Alzheimer's Association, in which the disease was given a clinical biomarker construct. The diagnosis of the Alzheimer's disease patients in the clinics continued to be done as having probable or possible AD, and the biomarkers use was proposed to support the diagnosis of the disease, although the confirmation of the disease could still be done only histopathologically. In 2018, the National Institute of Aging Alzheimer's Association, it proposed a research framework in which the disease was given a biological construct. The clinical symptoms were separated from the disease pathology, and it was recognized that the disease could still exist without the patient showing clinical symptoms. The disease could be identified in the living with the use of biomarkers using the ATN classification of biomarkers. This is the ATN classification, which divides the biomarkers into amyloidosis, tauopathy, and neurodegeneration category. The N category is placed in parenthesis because it represents neurodegeneration and is not specific for Alzheimer's disease. Each category consists of CSF and imaging biomarkers. So our research was was an effort towards finding early and reliable diagnostic tools for Alzheimer's disease in our population, because early diagnosis can lead to early intervention and hence delay in the onset of symptoms. The currently used biomarkers are either from CSF or they employ PET scans, which have limited utility in Pakistan, because in Pakistan we have very limited number of PET scans available and which are very expensive, and CSF analysis is invasive. So globally, the research is directed more towards finding blood-based biomarkers for the disease. And these three proteins in literature are evolving as potential and promising biomarkers for Alzheimer's disease. So we compared the mean plasma levels of A beta 42, P tau, and neurofilament light in patients who were diagnosed with neurodegenerative dementia with Alzheimer's clinical syndrome and their cognitively unimpaired healthy controls. For this, we included the patients who were diagnosed with neurodegenerative dementias and who consented to be a part of the study, while those with history of psychiatric disorders, brain injury, TIA or CVA, normal pressure hydrocephalus, and brain tumor were excluded. This analytical cross-sectional study took place in the neurology OPDs of Civil Hospital and Dow Hospital Ocha campus and A.Q. Khan Institute of Behavioral Sciences. The subjects presenting with neurodegenerative diseases who were diagnosed as having Alzheimer's clinical syndrome by the physicians were included in the study through non-probability convenience sampling. Their age-matched healthy controls were recruited from amongst the attendants from the same healthcare facilities. After informed consent, the MMSE score of all subjects was calculated and a questionnaire was required to be filled, containing lifestyle and demographic profiles of the patients. 5 cc blood sample was collected in EDTA tubes, transferred to lab in ice, and was centrifuged. After collection of plasma, the plasma was stored at -80 degrees Celsius until further analysis. The protein levels were assessed using ELISA techniques, commercially available ELISA kits, using the manufacturer's protocol. The plasma samples for each subject were run in duplicates. The statistical analysis was done using IBM SPSS version 22. Central tendencies were computed. Mann-Whitney U-test was done to compare these mean plasma levels. The ROC analysis was carried out to find cutoff values for each of these biomarkers. As depicted by this table, we see that both the groups of the ACS and the cognitively unimpaired patients were pretty homogeneous in terms of their age, gender, and comorbid profile, except for depression, which was found significantly raised in the Alzheimer's disease patients. The mean plasma levels of NFL and p-tau were significantly raised in the ACS group as compared to the control group. The A-beta 42 levels were found lower in the plasma of the Alzheimer's disease patients as compared to controls, but the difference was not statistically significant. The ROC analysis was also not able to differentiate the ACS group from cognitively unimpaired group in terms of A-beta 42, as is depicted by the p-value, which is more than 0.05. However, the NFL and p-tau, they were able to differentiate between the two groups with an AUC of 0.735 and 0.717 respectively. Correlation analysis also revealed that there is a significant negative correlation between plasma NFL and p-tau levels. So the patients who had lower MMSE scores had significantly raised levels of NFL and p-tau in their plasma. Surprising also to meeting the literature, 68% of Alzheimer's disease patients were women in our study, which corresponds with the globally reported figure of women having Alzheimer's disease. Our study proposes the use of blood-based biomarkers, which, if brought into practice, can improve the diagnosis, because this will be less expensive, more easily accessible, will require less expertise, and hence blood-based biomarkers are more practical for use, widespread use. However, there are some challenges with using blood-based biomarkers, some important ones being that these proteins, they have to cross the blood-brain barrier, and hence very low levels of these proteins appear in the plasma, which is difficult to measure. Dilution factor is higher in the extracellular fluid as compared to CSF, and also the protease action is more pronounced in plasma as compared to in CSF. However, our results are supported by other results in literature, which also propose the use of NFL and p-tau in differentiating Alzheimer's disease patients from healthy controls. However, the plasma A-beta 42 levels have shown inconsistent results in literature. A few reasons that we could identify were that the different techniques were employed to measure the protein levels in different studies, which could have accounted for the difference in results. Also, the difference in the diagnostic evaluations of the patients was observed. Some studies stratify patients based on their clinical symptoms, while some studies stratify patients based on their amyloid burden, either detected through CSF or through PET. Also, the different stages of the disease were used in different studies, ranging from subjective cognitive decline to severe cognitive impairment. To cope up with these differences and this blood-based biomarker personalized interest area has been convened by the Alzheimer's Association, which is working towards standardizing the use of blood-based biomarkers for Alzheimer's disease. Recognizing the limitations of our study, the small sample size of our study limits the generalizability of our results. Also, the selection of subjects was based on the diagnosis of the attending physician, which could have varied from physician to physician. Also, the study was not blinded, and the investigators carrying out the analysis, they were aware of the diagnosis of the sample. These are the references of the study, and thank you so much.
Thank you so much, Dr. Tanya.
Thank you so much, Dr. Thaniyat. Next, we would like to invite Professor Dr. Aisha Ishaq to give her speech. either way. This is the pointer as well as the changing, and this is the time, so you can see how long.
Okay, and how much time?
5 minutes.
5 minutes, okay. Presentation is not. Okay. दे दी थी presentation. Okay, Auzubillahiminash-shaytanir-rajeem. Bismillahir-rahmanir-rahim. Assalamu alaikum everyone. I'm Aisha Ishaq, PhD Research Fellow at Dr. Panjwani Center for Molecular Medicine and Drug Research. The presentation that I will deliver today entitles the mesenchymal stem cells for the treatment of neurodegenerative disorders. As you all know, the regenerative medicine is mainly concerned with the integrity of the organs, and it mainly aims for the regeneration of a particular organ. In the context of neurodegenerative disorders, USB, yes, in my bag. Okay. So the title of my presentation is Mesenchymal Stem Cells for the Treatment of Neurodegenerative Ailments. What are the neurodegenerative disorders? Neurodegeneration is actually a gradual, prolonged and selective phenomenon of the neuronal damage that affect the CNS badly. And it significantly impairs the neuronal functions as well as the structural integrity of the brain, and which results in the brain atrophy and the overall structural distortion of the brain structure. Such anomalies have still not been identified for any proper treatment options. So in my case, the disease that I am targeting for the treatment option is a cerebral ischemia, which is also termed as a stroke. The stroke is actually the hypoxic condition which can cause the degeneration of the neuronal cells within the brain. The stroke can be this, locally or the globally damage of neuronal cells, which hardly affect any certain body parts, and thereby results in the paralysis and other symptoms associated with the ischemic stroke. So what is the body's natural response against the stroke? We have neural stem cells within the brain from the SVG, which is the along with the SVG, we have the VGS system, which where the neural stem cells exist, and at the time of injury, the stem cells becomes activated and differentiated towards a particular cell type and can perform the regeneration of this particular disease. So the treatment strategies, currently there are not many effective therapies available that can treat with the particular degenerative disorders. So therefore, we are aim and we are hopeful to treat the particular neurodegenerative disorders of any kind by using the stem cell therapy. This is called the regenerative medicine, by which the particular stem cells are used for the treatment of neurodegenerative disorders. And how to use the MSCs? We can use MSCs in their native form by isolating them naturally from the body and also by preconditioning them. And what is the preconditioning? This is a phenomena of enhancing the regeneration potential of the stem cells by using the different genetic manipulation, or we can use the particular compounds, which are the chemical factors which can induces the differentiation or enhances the therapeutic potential of stem cells, and these are responsible to treat the particular area or the damaged body parts. And how do the stem cells undergo differentiation? It actually, whenever we aim to precondition the stem cells, it basically hit a particular pathway, and this pathway leads to the differentiation of stem cell towards a particular cell type. In this case, we are differentiating the stem cells towards the neuronal lineage, and for this purpose, we have used the chemical compounds of class monoterpenes, which was utilized to perform the differentiation of stem cells towards the neuronal cells. So cell-based therapeutic approach for the neuronal regeneration in the stem cell laboratory of the Dr. Panjwani Center for Molecular Medicine. We are performing both in vitro and the in vivo work. For the in vitro work, we perform the differentiation of mesenchymal stem cells, and the stem cells are isolated from both bone marrow as well as from the umbilical cord. And for the in vivo work, we perform the transplantation of these differentiated stem cells into the rat model. And these transplanted stem cells are responsible for the regeneration of the defected or the degenerated body parts. The biomolecules that we have selected for the differentiation belong to the monoterpene class, and these compounds have already been reported by our group for the neuronal differentiation potential. That's why we use this compound for the regeneration of the damaged brain area as a result of the ischemic stroke. This is the study design that we performed for our study. The in vitro work, which comprises of the cells isolation followed by the characterization of MSCs. When this was confirmed that the cells are MSCs, we performed the cytotoxicity analysis to confirm the toxicity of the cells, and then we performed the neuronal differentiation. When the cells were differentiated, they were transplanted into the rat model. And once they are transplanted into the rat model, the regeneration was verified at the multiple parameters, which include the transcriptional approach, histochemical approach, histological approach, and etc.
Aisha, can you please wrap up?
Yes, within 30 seconds. This is the protocol how we isolate the stem cells from the bone marrow. And these are the cultured cells indicating the morphology of the stem cells at the different time of isolation. After the isolation, we perform the characterization of MSCs by using the immunocytochemical analysis, flow cytometry analysis, and tri-lineage differentiation potential. This is the cytotoxicity analysis, which give us the safe concentration of compounds. And these are the results of neuronal differentiation. When we use compounds into that particular stem cell, this helps to differentiate these cells into the neuronal cells. Gene expression analysis indicating the proper differentiation of cells towards the neuronal lineage. And these are the different markers indicating the neuronal lineage. We also determine the signaling pathways that are responsible for the differentiation of MSCs towards neuronal cells. Protein expression analysis of the differentiated cells. The cells were positive for the GFAP, NUN, tubulin, and synaptophysin. This is the model establishment, how we develop this model. This is by the 2 arteries ligation model. The model was also checked by using the assessment of brain by the TTC staining. This one is the normal control comparing to the degenerated model, which indicates the whitish area followed by the damage. And these are the treated groups, which indicate the regeneration of the brain after the transplantation of MSCs. These are the histological analysis indicating the regeneration of brain area after the transplantation of MSCs. Transcriptional analysis indicating the downregulation of inflammatory genes after the MSCs transplantation. NUN staining indicating the differentiation of cells after the transplantation within the brain region. And in conclusion, the regenerative medicine provides therapeutic potential for the neurodegenerative disorders, and this was the aim of our study. Thank you.
Thank you.
Thank you so much. Now I'd like to invite Rabit Tariq onto stage to finish up her presentation.
Thank you. Is this open? Hi. My name is Rabit Tariq. I'm a final year medical student from Liaquat National Hospital. And the topic of my presentation is about transcranial direct current stimulation in Alzheimer's patients. And these are my co-authors. So we received no funding for this project, and we also have no conflicts of interest to declare. For an introduction, there have been many very honorable speakers who had introduced to Alzheimer's disease. I'm just going to say it's the most common form of dementia we have, and we have pharmacological treatments for this. But since it's a non-reversible disease, we do not have any agent which reverses the disease. We only have agents to make the cognitive status better. There are several drugs in trial, which like monoclonal antibodies, which are extremely expensive, especially for LMICs. So there's this. So we have non-pharmacological agents such as transcranial direct current stimulation, which is also in trial, and which is comparatively a much better option for LMICs and a much cheaper option as well. So transcranial direct current stimulation is a deep brain stimulation technique. It's pretty similar to magnetic stimulation, except that we have anodal and cathodal stimulation. So depending on the type of stimulation that we want, we place the anode or the cathode on the place above the scalp, wherever we want to stimulate it, and we make sure that we have a current running over it. So the objective of our study is to assess the effects of transcranial direct current stimulation in the Alzheimer's patients, including effects on cognitive status, memory, attention, language, IQ, depression, neuropsychological statuses. We also assessed the factors that influence the outcomes. So we use the PRISMA guidelines. This is our PRISMA flowchart. So these are the results of individual studies. So for the outcomes, we categorize the outcomes into general cognitive status, memory, executive function, language, orientation, etc. To summarize my results, so basically, there were very heterogeneous results with only 33% studies reporting an improvement in the general cognitive status. And there was almost no improvement in associative memory, only recognition memory was improved in half of the studies. Attention span was also not improved. Cathodal stimulation may improve fluency, verbal fluency, and anodal may improve naming abilities. But there were no major adverse effects. Working memory may be improved by multi-session as well. So you can see there are many heterogeneicities in the studies. So we analyzed why were the studies reporting so many differences. So the factors that influenced the outcomes were stimulation polarity and the areas which were stimulated. So anodal stimulation basically causes depolarization, and cathodal, on the other hand, causes hyperpolarization. So anodal may be effective for cognitive enhancement, and cathodal doesn't have any effect, or it might be the opposite as well. For the area stimulated, we were expecting an effect on the verbal memory as a result of left temporal cortex stimulation, but we didn't observe that. There was an improvement in overall cognition by DLPFC stimulation, but that was only in 28% cases. However, bilateral temporoparietal stimulation resulted in improvement in several cognitive domains. Trainings given and the functional nervous state were some of the confounding factors, with those with MCI and a better resting state having a better result. So basically, these were the instruments that were used for the assessment, and since there were a lot of instruments used by different studies, we could not actually analyze them. We could not do a meta-analysis or a quantitative analysis, which is one of the limitations of studies. Another limitation is a lack of, some studies did not provide enough data. The conclusion is that since it's a new technique and it's a very feasible one, there has to be more studies considering these factors which I had mentioned, that there has to be a proper brain mapping technique to identify areas of stimulation, and then obviously anodal stimulation is more needed than any other kind of stimulation. So we have more studies which are warranted for a better understanding of the therapy. This also, the final acknowledgement is this study was conducted by a multinational collaboration of International Society of Chronic Illnesses. So we have collaborators from India, Nepal and Pakistan who had worked on this, and thank you so much.
Thank you.
Is this your phone? Thank you, Rabit. Next, in a few moments, we'll be hearing a speech from Dr. Soeba Jawed. It's going to be about cognitive and neural engineering. It will be another virtual presentation, so I'll give you guys a few minutes to set up. It is our keynote address from Dr. Soyaba Javed. Can you hear us?
I can hear you. Can you hear me?
Okay, thank you so much. And whenever... Okay. You can get started when you're ready. We can hear you as well.
Presentation. Can you see my presentation?
Sweba, we cannot see your screen. Oh yes, now it is here. Please proceed. Sweba, can you hear us? Please start because we only have 10 minutes for this.
Hello, can you hear us? You can start whenever you're ready.
I am ready. I wanted to ask you, can you see my presentation?
Yes, we can see your presentation.
All right, awesome. So today talking about assessment model for identification of visual learners using EEG signals. This audience is, I believe, all medical students. So I will take you to another space where you can see how you can utilize EEG modalities or neuroimaging modalities for non-medical purposes. So I have introduction, then I'll be talking about methodology, and it will finish with a conclusion. So in my idea of future of learning, learning adapts to each and every person. Now this, because I think every person exactly has what it needs to be successful. This vibrant feature of learning is possible, but it is not a norm these days. That's why where the ideas of personalized learning come in. So if we look at the statistics, according to 2010, the completion rate of undergraduate students is just 29%. And these all undergrad students were taught using conventional methods. And personalized learning is basically the approaches which is based and develop to tell them how they can learn better. So in literature, there are many existing learning models, naming few are Felder and Silverman, there is Karanian model.
Soiba, this is Athar Enam.
Yes.
आपकी slide नहीं आगे बढ़ रही है. Your slide is stuck.
नहीं आगे बढ़ रही.
Right. Are you seeing it on your own screen? So you may have to.
I can.
You may have to switch it off and then switch it on again, I suppose. And if that doesn't work, then log off it, then log on. So in that case, we may have to extend the time.
So I should. Okay, I will. Now can you see it?
No, still stuck on the first slide. Any issues from there? Do you want to unshare and then share again? That's what, right?
Yes, that's what I'm going to do.
So log out is an option, but then that will take more time.
Yes, and I think the next speaker is also.
So now share again.
Can you see it now?
Not yet. Not yet. Yeah, okay, we can see it now, but then go ahead, move it forward. आगे जाएं. Slide number 2 दिखाएं.
देख रहे हैं सर आप अभी? Can you see it?
I'm still seeing the slide number 1.
Okay. I don't know how teams work normally.
Well, that's exactly like my sister. My sister is also a computer scientist, and she has a B from NED University, and she does the same thing always. So, you know, doctors have better understanding of this. Okay, now we see the second slide. Good, excellent. Thank you.
All right, and then I stop sharing. 1 minute, I'm sorry. Browse from my computer. Okay. I think it's a delay because of the internet. Can you see it now?
We can see the first slide, Dr. Soiba.
I think you need to wait a bit. Maybe it's the internet connection. Now, is it moving?
No, I don't see anything, Dr. Soiba.
Not even the first one?
So if I do this, you just come on, why don't you stay over there, and we will run the slides from over here. How about that? You just tell us next slide.
That's a very good idea. Yeah, I can send you the slides.
Huh?
So, shall I email it to you?
Yes, so let us put the slide over here. We will hear your voice.
That would be nice.
Okay. Just say next slide. We will start a control from here. Just say next slide.
All right, so I shall email you the slides first. 1 minute.
We have the slides.
Yeah, we have the slides. You just say next slide.
Oh, all right, awesome.
Okay.
So I am on the 4th slide. I'm on the 4th slide, including the introduction, including the title, which says learning models.
Okay, we are seeing that second slide. We're talking about learning models.
Yes. So there exist many learning models which can identify the learning style of the students, but there is a lack of theoretical coherence and common framework which is found by researchers among these models. Next slide. But all of the researchers, they agree on 4 learning modalities. So you could be a visual learner, you could be an auditory learner, you could be a tactile learner, or you could be a kinesthetic learner. So if you're a visual learner, you learn through seeing things, watching videos or visuals. If you're an auditory learner, you learn by hearing things, you know. And if you're a tactile or kinesthetic learner, you learn when you do things, you perform things using your hands. So although a student can have one of these 4 learning modalities, next slide, 70% of the students are visual learners and audio learners are 20%. The irony is 80% of classroom lectures are delivered orally. So it's very important to identify the learning style of the student, but we need to see the brain patterns, because if you tell someone learning style subjectively, probably it's a learned behavior, and that will increase the cognitive load. So for brain patterns, we can use EEG because it is very easy to take it to the classrooms, and it is cheap, also readily available. So the idea is to identify the learning style in the classrooms to improve the fallout rate of the students. And then these are many other learning modalities, because all of you are medical doctors, probably you know them better than me, but we use EEG. And the idea behind this is there are existing objective studies, but their accuracy is very low. And also the other limitation is you cannot use them in real time. So I will like to tell you about real time. By real time means that you can record while someone is learning and you can process it at the same time, and then you can simply tell them the output. So this work has not been done yet. So these are the 2 things that's why we try to address this problem. And how we do it? So we collect the EEG data while students were learning, and then we extract the features, and then we classify it into their learning style. So the data was collected from 34 subjects, and it was 2D learning and recall. And I'm sorry, I stopped saying next slide. So the EEG data, it's slide number 11, by the way. So it's EEG data which is collected from 34 subjects, and they were learning, and the things they were learning are animated learning content, which is of 8-minute durations, and the content of animation were human skull and its structure, brain anatomy, and brain disorder, Alzheimer's disease. And once they learn, then they take the recall test, which is consists of a multiple choice question, and they answer the question. The next session actually happen after 2 months, and then another session after 2 months, and then another session after 2 months. So we see that who retain the information well. And then we take this, we clean the data, and we put it into a machine learning pipeline. We extract the feature and we classify the student into a visual learner and a non-visual learner. And at the same time, what we do is we take the raw EEG data and we give it to the deep learning pipeline, and then we classify and tell if someone is a visual learner or a non-visual learner. So this is the block diagram, how we do it. So when we talk about machine learnings, we need to basically divide the data into test and train. So you train on some data and then you test it on the rest of the data to see how well your model is working. So we take the data, we extract the feature, and then we classify it, and then we test the data, and then we use ROC curves and confusion metrics to see the how good the model is. And I concluded my presentation with these things. So basically there are 2 steps. So we collect the EEG data from the students who were learning, then we pre-process the data, extract features, and then classify into 2 groups. In the second phase, we use deep learning models, and we classify the students into visual learners and non-learners. If you have any questions, you can ask me.
Thank you so much. Thank you so much, all of you, all the physical participant and the virtual participation. We are directly proceeding towards our next session, which is the last session of today. The tea break will happen after the next session. For question and answer, all the student and attendees are encouraged to directly connect with the presenters, and I hope that you will have a fruitful discussion with them. Thank you very much.
Musa is doing. Thank you so much, Dr. Bushra. I would like to do an audio-visual check before we start news. Dr. Zafar, Professor Emma, can you hear me? Can you hear us? And can you please start sharing your screen if that is possible, so we can start with news as soon as possible?
I can hear you clearly. I can hear you. I just need to sort out my screen share.
Good morning, good afternoon. So we'll be starting news shortly. I would like to introduce the moderator for today's session, Mohammed Musa Hamza, who's a second-year medical student and also the part of NSIG.
Good afternoon, everyone. To all our guests, speakers, and everyone watching, it is my distinct honor and privilege to welcome you all to another session of the Neuroscience Educational Webinar Series, or as we call it, the News, presented by Pakistan Society of Basic and Applied Neuroscience. I am Mohammed Musa Hamza, and I am currently a medical student at the Aga Khan University, and I am a member of the Neuroscience Interest Group. News, or Neuroscience Educational Webinar Series, is a program held on a monthly basis in which researchers and neuroscientists from all over the globe talk about the new advancements in the field of neuroscience. However, today is a special session of news, since it's the first time we are conducting this session in person as well as broadcasting it online. Today we have a special guest for our session, Professor Dr. Emma Robinson, and the topic for today's session is investigating the neuropsychological effects of antidepressants in a translational rodent model. The webinar today will begin with a 45-minute presentation followed by a 15-minute question and answer session, and then there will be a 20-minute general discussion. You all are encouraged to ask questions during the question and answer session only. If you have any queries during the presentation, please write it down for later. During the Q&A session, if you have a question, you are encouraged and requested to raise your hand. For people who have joined us online, please only unmute yourself when I call your name out. You are also requested to turn on your camera so that we can make this an interactive session. You can also send in your questions in the chat box as well. I would like to emphasize that for the certificates, it is essential to fill out the feedback form, and the link for that will be provided at the end of this session. Today we have with us many brilliant minds joining us from Pakistan and hopefully around the globe. We are joined by our director of the Neuroscience webinar series, Professor Zafar Bashir. Dr. Zafar Bashir completed his PhD from the University of Glasgow. After 2 postdoctoral stays in the University of Bristol and Birmingham, he got a Wellcome Trust Career Development Fellowship in the University of Bristol, where he established his laboratory. At present, Zafar Bashir is a professor of cellular neuroscience at the University of Bristol. The focus of his lab is to assess the role of glutamate receptors in synaptic functions and plasticity, using the hippocampus and the neocortex as models. He has made key contributions to understand the role of metatropic glutamate receptors in long-term depression. Dr. Zafar Bashir is also the chair for this session. A round of applause for Dr. Zafar Bashir. I would like to ask Dr. Zafar Bashir to please say a few words.
Okay, thank you. Thank you very much. Hopefully you can hear me okay. Thank you very much for the kind introduction. So as was just said, I'm chair of the news seminar series. So the annual Neuroscience conference is a fantastic, I've attended this online a couple of times now, and it's a brilliant meeting which brings together people conducting various neuroscience work across Pakistan, and it's a fantastic way of hearing about the neuroscience that goes on in Pakistan. When having discussions with Dr. Athar Anam, we were thinking about ways that we could actually broaden neuroscience in Pakistan to a wider audience and bring in speakers from outside as well. And so we established the news seminar series, which started in January this year. So this is our 10th news seminar series, and it's great to have Professor Emma Robinson. I'll leave the introductions for the speaker for a little bit later to somebody else. And so this is the 10th of our seminar series. And so these happen on the first Wednesday of every month. And it was either a pure coincidence or very good planning by somebody that actually the first Wednesday of the month of our seminar series coincided with the Pakistan Annual Neuroscience Conference, which means that our seminar could be held in association also with the Annual Neuroscience Conference. So as I said, we hold this first Wednesday of every month, and we have speakers from around the globe, and we've had a good range of different speakers speaking on all sorts of topics, from sleep through to generation of cells in the dentate gyrus and the function of those cells, for example, and a whole range of other topics as well. So I'd encourage everybody to look up the news, and you can find the website from the PASBAN site, the Pakistan Society of Basic Neuroscience site. So if you go there, there's a link to the news webinar series, and you can also find there all the past sessions, which are all recorded as well. So the sessions are all recorded from before, but if you want to join us live, you can on the first Wednesday of every month. The next one will be on the 2nd of November. So as well as myself and Dr. Athar Anam, there's a steering committee that helps organise speakers and suggest speakers. And we also have a logistics committee that actually does all the work that makes the seminar series happen. So I'd just like to say a big thank you to those people as well who help make the seminar series happen. So I'm going to stop talking now. This is not about me. This is about our seminar today. So I'm going to hand back now for the introductions for our speaker today, who's Emma Robinson. So thank you very much.
Next, our esteemed speaker, Professor Emma Robinson, that I have the honor of introducing. Professor Emma Robinson is a professor of psychopharmacology at the University of Bristol. She completed her degree and PhD at the University of Bristol, where she was awarded a fellowship in 2005, and after working at the University of Cambridge in experimental psychology, she returned to Bristol to establish her own research group. The focus of the lab is understanding the mechanism which regulate cognitive and emotional behavior. Work in the lab uses behavioral studies alongside neuropharmacological and neurochemical approaches to study the role of specific neural and neurochemical systems in the control of behavior. But in short, let me just say that Professor Emma Robinson, we are so honored to have you here with us. And let me start by thanking you for taking out your precious time for this session. It is truly remarkable for how much you have achieved. And I'm sure I, along with other participants over here, are looking forward to a presentation. Over to you, Dr. Emma Robinson.
Thank you very much, and thank you very much for that introduction. I'm just going to check everybody can hear me okay, and that my slides are moving. Are they moving forward?
Yes, we can.
Yeah, okay, that's great. So hopefully all the technology will work. So I'm going to talk to you today about an area of research that I've been involved with for the last sort of 15 years, and it relates to trying to understand more about depression and the treatments that we have for depression. But in particular, it builds from work we've been doing developing a translational rodent model. And that has been, I think, very important for what we've been able to achieve, because we've been able to take these a model system and actually ask important questions about how these drugs may be working, and then sort of gain some mechanistic insight. So I'm going to tell you a little bit about the kind of background and then some of the more detailed experimental work that we've been involved with. So I'm sure I don't need to remind this audience or tell this audience, but I always think it's good to just sort of give some context to the disorders that we're interested in studying. And whilst I spend most of my time working with rodent models, actually, you know, I always try to keep in mind the clinical relevance of what we're doing. And as I'm sure you're aware, major depressive disorder is one of the biggest challenges we face in the 21st century. It is seen as a major problem in terms of long-term disability in our population. It's thought to affect somewhere in the region of perhaps 300 million people, and there's a very significant number of people who die as a result of the disorder, linked particularly to suicide, but it's also associated with a lot of other diseases. And so there's a link to addiction, obesity, chronic pain, neurodegenerative disorders. These are all disorders where we see changes in mood as a component of those, and one may drive the other, but actually there may also be these comorbidities where the outcome for patients with depression and these conditions is much worse. The other thing that I think is really challenging about this disorder is that, you know, we talk a lot about stress and we talk about the kind of links and, you know, early life adversity and these potential factors that cause depression, but actually we still don't really know why it develops. And nobody can even really agree about the antidepressant drugs. So there's been lots of articles around whether or not antidepressant drugs really have got beneficial effects, have efficacy, and there's a lot of concern amongst the general public that they don't necessarily have all the information they need to make decisions about whether they should or shouldn't be taking antidepressant drugs. So it's a really quite confusing picture. So just very briefly, I mean, we have a very interesting history of treating major depressive disorder. So back in our early history, it was linked, and certainly a kind of European approach was seen as this was a spiritual disorder linked to demons and evil spirits. And Hippocrates talked about melancholia, excessive of black bile in the spleen, and these kind of ideas, but in 1621 that Robert Burton's Anatomy of Melancholia first actually started to introduce some of the concepts that we might be very familiar with today. So the importance of social and psychological factors in depression, and that's what I've always been really interested in. That I think, you know, we often try to look to a very sort of try to refine this to a very basic biological cause, but in dealing with these psychiatric symptoms, we perhaps sometimes don't fully recognize the importance of these social and psychological factors, and certainly in my field of animal-based studies that's been the case. We've had various different types of treatments that people have tried, but in the 1960s was when there was a big change. We saw the introduction of pharmacological and psychological treatments. So we had drugs like imipramine that had been found serendipitously to have effects on mood, and also we had people like Beck proposing this idea of a cognitive theory of depression, where rather than looking at the biological basis, this was very much focusing on how people felt about themselves, the world and the future, and this feeling that hopelessness formed, and you got this sort of triad of detrimental effects. My apologies, I have a bit of a cough, so I'll try not to keep coughing through this talk. And I think it's also very interesting that we still base a lot of our discussions about depression around theories of depression that have not ever been really fully substantiated. And so the most popular of these is probably the monoamine hypothesis. And this is based really on the mechanism of action of conventional antidepressants. And because these were discovered serendipitously, we then later found out how they work. So we then discovered that they had actions on the monoamine system, and then we made an interpretation of that perhaps they were reversing a deficit in those monoamine transmitters. But in fact, there's never been any evidence of a direct deficit in any of the major transmitters, including serotonin, which is our primary target. And in fact, there was recently a very important review by Moncrief. It suggested that, you know, this idea that there was a relatively simplified down biological basis to depression, too little of a transmitter, was not actually the case. And I think it's really important that we recognize this, because actually how people perceive their disorder has major impacts on their outcomes. So if people are told that it's a purely biological cause, they often make very little effort to change aspects of their life, which could in fact be beneficial, and the outcomes for those patients can be worse than those that recognise the importance of psychological factors. The neurotrophic hypothesis arose really when we couldn't fully explain everything based on monoamines, and particularly around delayed onset of action of antidepressants, the conventional antidepressants. And this was based around the idea that stress, and this was very strongly supported by some work in animal models, that stress could induce atrophy within brain regions, and that those brain regions were important for the modulation of emotional and cognitive behaviours. And we could see this very nicely by doing imaging work in animals which were exposed to chronic stress, and there was links to the monoamine transmitters and even the glutamate transmitters in terms of how they could modulate the synaptic processes. Oh, Zaf just brought me water. Thank you, Zaf. That's very kind of you. And so if we think about the stress-induced atrophy, then how do antidepressants interact with that? And there was some really nice evidence from, again, the animal studies showing that antidepressants via probably brain-derived neurotrophic factor signaling could reverse the loss of synapses and rebuild these connections. And so really the literature has been strongly dominated by these two hypotheses. But the area I've really been interested was a perhaps somewhat quieter group of researchers who were working on a slightly different approach to this. And rather than building from a purely biological cause, they were interested in neuropsychological mechanisms. And this was suggesting that, in fact, that there may be a relationship between the biological processes and psychological factors, and it was the integration of these and the combination of these factors that could be particularly involved in depression and in antidepressant efficacy. So before I talk about that in detail, I'm just going to deviate for one moment into this area of how we actually study these behaviours and these symptoms. So I think when I first came into this field, one of the biggest challenges I could see, and this was when I was a PhD student, one of the biggest challenges was how do we actually quantify these subjective kind of self-reported feelings associated with depression? How are we going to measure those in animals and be able to do this kind of translational research? And in fact, I think perhaps this is a challenge I would set to the clinicians as well as to animal researchers. Is subjective self-report the best way of us fully really being able to quantify the symptoms of depression and be able to even understand the response of patients to antidepressants? And when it comes to animal models, at the time I started my PhD, we were very limited. So we had things like sucrose preference, where we looked at the hedonic response of animals, their reward sensitivity, and we aligned this with this idea that over here we had symptoms where people had a loss of interest. But this was a fairly anthropomorphic type of, alignment, and we didn't actually ever really test, do humans with depression have a change in their sensitivity to sucrose? We also use models like this where we have this, looking at anxiety-related behaviour. But the reality is it's very difficult to use these approaches. However, if we think about the relationship, the neuropsychological hypothesis, then what emerged for me was the potential to actually develop translational methods. So here we have affective biases in depression. Now this is where we've got really quite good evidence that when people are experiencing depression, this emotional state can bias their cognition and executive functions. So what we mean by that is that attention, learning, memory, decision making, I think we've all probably experienced it. It's something that, as our affective state, our emotional state changes, we interpret our world differently. And that is very normal. But in people with depression, what's become very evident is that they have this pervasive and persistent negative affective bias. So this negative affective biases seem to have this long-term impact on all these different aspects of cognition and potentially then impacting on mood. What was very exciting was there was some research that showed that conventional antidepressants could induce an acute shift in these emotional processing type tasks, but subjective effects required psychological factors for them to actually have a subjective improvement for the patient. So this really is where the neuropsychological hypothesis of antidepressant efficacy developed, and the idea was that there was the potential for the pharmacological treatments to be sort of setting a biological environment. But those alone were not sufficient to give clinical benefit. What you needed was social and environmental factors. So what was also really important about this development in the field was that this was not a self-report measure. This was something that could be quantified using a computer-based task by looking at cognition. So you could study cognitive processes in patients and look at how they differed between patients and controls, and you could see what you could attribute to be an affective bias. And the same thing could essentially be done with animals. So we, along with others, developed tasks where we were using these same cognitive domains. So we could ask animals to make decisions, or we could ask animals to undertake learning and memory tasks. And we could look at how affective state biased those behaviors. And as such, we could provide a quantification, an actual, put a number to that affective bias. And there's two tasks that we've been involved with. The judgment bias task, which looks at decision making. And this is where animals can be trained to respond to cues that predict reward or predict less positive outcomes or aversive outcomes. And then we can probe their behavior using intermediate cues. And we can say, are you a glass half empty, glass half full kind of rat? I'm going to talk mostly, though, about some work here which we've focused on, which is the affective bias test. And this is a task that I developed to look at reward learning and memory, and it's based on a very simple foraging behavior in rodents, very easy to train. It does require a lot of manual interaction and handling from researchers, but it's given us some really exciting data. Now, just before I go on to talk about the actual data, I want to introduce one other aspect, and this is kind of one of the most exciting things I think that's happened in psychiatry, but also it's posed a lot of challenges to our hypotheses about how antidepressants work, and it's meant we've had to, and certainly for me, I've had to really rethink some of what I thought was established understanding of antidepressant drugs. I always thought that delayed onset of action was just part of how depression developed and then how depression was treated. But in 2000, this very exciting paper came out showing that ketamine, a drug we've traditionally always known of as a drug for pain management, anesthesia, at very low doses, it had this potent and rapid antidepressant effect. So this graph at the bottom here, this is what we would consider the kind of dissociative aspect of ketamine. So this is when people feel that they have the drug is causing some sort of dissociation. But that's very, very short-lived. And in fact, the antidepressant effect is seen much later and is sustained. And the really exciting finding was that after a single dose of ketamine, the effects of the people's mood reversed, but it reversed and was sustained long after the drug had left, and could last for many hours, for many days. And in some patients, this can go up to 2 weeks. The other thing that was very exciting is that it was also anti-suicide. It massively reduced suicidal ideation. And it could really be seen as a drug with the potential for sort of emergency care in psychiatric patients. And it's not just the ketamine, it's subsequently, we've seen a huge number of trials and excitement around the potential for a whole range of different drugs that have been shown to have these rapid-acting antidepressant effects. Interestingly, they're also nearly all drugs of abuse, but substances like MDMA or ecstasy, the psychedelics, so drugs acting on the serotonergic system, particularly psilocybin and LSD, which of course LSD in the 70s was used for drug-assisted psychotherapy, so there's quite a precedent. But what we're now seeing is clinical data showing that these drugs have rapid-acting antidepressant. And an interesting drug as well, which is scopolamine, which is a muscarinic antagonist, more commonly known for its association with crime in countries like Colombia, where it's blown into people's faces, called devil's breath, and it causes this amnesic effect. But at low doses, it's been shown to be antidepressant. So we have a really exciting but also lots of questions that this raises. How is it that our established ideas of delayed onset of action and these conventional antidepressant drugs working through things like monoamine reuptake now, and possibly through neurotrophic mechanisms, and now we've got these drugs that seem to be able to reverse these symptoms immediately. And then for a pharmacologist like me, one of the bizarrest things is that the effects are sustained, and their effects are sustained long after the drug has been metabolized and excreted. So I've talked about the limitations of animal models, so I'm not going to repeat that. But what I just want to really emphasize is the problem we have is that subjective symptoms in patients do not equal behavioral deficits in animals, and we have to make these interpretations, and that can be problematic. But when it comes to objective changes in affective cognition, this is something we can model in animals, and this is what we've done. So the assay that I'm going to show you data from, and this is the sort of the basics of how the method works. So this is built on the idea that patients with depression attribute less value to positive experiences. And those of you who have been involved clinically in working with these patients, but also many of us have experienced patients, have experienced friends or relatives that have depression. And it's something I'm very aware that you talk to these people and they've been out and had the same experience as you've had, but when you talk about it, their memory seems to be much more negative about it, or certainly less positive than yours. And I wondered whether we could model that really quite simple idea if in animals, if we generate a hypothesis that affective state at the time a positive experience is encountered can bias the memory, and that when you subsequently retrieve that memory, that is, if in people with depression, that reward value is less. And we devised an experiment that would enable us to test that, which is actually really relatively simple. It's a cue reward association task, and it involves animals being trained to discriminate between digging substrates. So rats are very, very good at this. They can very quickly learn that this digging substrate has a food reward in it, but this doesn't, and then they will repeatedly return to this substrate to dig to find the food reward. So what we can do is we can do pairing sessions, so we can teach the rat that substrate A has the food reward in it, and then on the second day we can teach them that substrate B has the food reward in it. We can keep that food reward exactly the same amount, so one reward pellet on each occasion, and then to test our hypothesis that affective state can change the relative value that the animal remembers about that reward, we can manipulate their affective state before one of the learning experiences but not the other. We repeat the learning, and that's because we don't want the animals to just base their decisions on recency, and we find that if we do just 2 pairing sessions, one of each, we will get a recency effect. But if we do the 3rd and the 4th, then we see no recency effect. We repeat the effective manipulation and the pairing, the control manipulation and the pairing, and then in answer to ask the animal, which experience do you prefer, and i.e. what is your effective bias, what was the consequence of this effective state manipulation on your relative valuation of these experiences, we can use a simple preference test. And so we can present the animal with the two previously rewarded digging substrates, randomly reinforce them, and run 30 trials. And what happens is that if animals have been put in a positive effective state, they should bias towards A, and if they've been put in a negative effective state, they should bias towards the control. And we can use this task in several ways. So we can look at how we can bias new learning, and that's been particularly important for us in terms of hypotheses about how conventional antidepressants work. But also, we're really interested in how rapid-acting antidepressants work. And as you'll see from some of our early data, we didn't see effects in terms of new learning. But so we developed a protocol where we establish a biased memory. So we generate a negative affective bias, which should, under control conditions, will generate a bias away from the substrate learned under the effective state. But in order to see if that can be manipulated, can that be altered, we pre-treat here. So we give a drug either 1 hour or 24 hours before we do this retrieval, this preference test, before we ask them to retrieve the memories. So that's the basic protocol that we use. And then these are a set of data. So this is a huge amount of different dose response curves that we've done, and lots of different students' research. So let me just run through. So what we've got here is essentially, in all these studies, it's the same animal being asked to evaluate a control memory versus the manipulation. If the bias is above the line, then it's a positive bias. If it's below the line, then the animal has a negative affective bias. We have one positive control that we use, and this is a reward-induced bias. So in this situation, we gave them 2 pellets on one of the learning experiences and 1 pellet, and so we show a proof of concept. So value of reward will generate a bias. But all the other data you're looking at, these are all from studies where we just had a single reward pellet, and all of these studies had vehicle control experiments included in them, and so we know that the design works, and under a vehicle treatment, there is a 50-50, they come out with no bias, and then we see a dose-dependent bias, and the data I'm showing you here is the ED50. It's the dose that gave the effective concentration. And so we see this for conventional antidepressants, so fluoxetine, reboxetine, venlafaxine. These are reuptake inhibitors and a receptor blocking antidepressant. These induce a nice positive bias. So just to really emphasize the importance, the animal has only had exactly the same value food reward throughout. So this is being generated by the drug treatment, which we would argue is that it's generating a positive affective bias. We also are able to see the opposite. So drugs which are pro-depressant in humans, so these are drugs we know are associated with negative psychiatric symptoms in patients. So rimonabant, a CB1 antagonist, retinoic acid or roaccutane, a drug used to treat acne, and FG7142, which is a benzodiazepine inverse agonist, so that's an anxiogenic type of drug. These all generate a negative affective bias. Again, this is a within subject design. This is not a change in the animal's core affect. This is a reflection of how that specific learning experience has been biased. And we've also looked at stress and inflammatory markers. But I just want to highlight a couple of important pieces of data here, and that relates to the use of psychosocial manipulations, not just pharmacological. We wanted to really test the validity of our task. And in most previous animal studies, the task, the behavioral readout has limitations. So it works in some situations, but not in others. So for swim test, very good for predicting antidepressant drugs, but it's not sensitive to many of these pro-depressants, and it's got limited validity in terms of some of these models, and certainly the inflammatory models. But we found very nice effects with social enrichment. So animals were given a social play area for one of the learning experiences and just their control home cage for the other, and we see a positive bias. And then this is a 10-minute restraint stress. But I think it's also important to highlight that ketamine, this rapid-acting antidepressant, did absolutely nothing. So we saw no effect when we gave it in that kind of learning paradigm, which is in itself was really interesting. So it showed a dissociation between conventional and rapid-acting antidepressants. So we went on to ask the question, so could it be that in fact there is a neuropsychological basis to the difference in how these drugs are working? So we hypothesized that drugs which have a delayed onset and the conventional antidepressants will be working through effects on new learning, whereas the rapid-acting antidepressants may be able to modulate these previously learnt and biased memories. And that's exactly what we found. So to test this is using this protocol where we induce a negative affective bias, and then we give the drug treatment before the preference test. And so here you can see the expected negative affective bias. The conventional antidepressant venlafaxine had no effect. It failed to reverse that. However, ketamine did, and ketamine nicely reversed the effects. We also see the same with the psychosocial stress. So again, it's not just a pharmacologically induced negative affective bias, but also a stress-induced bias. And these are work we published some years ago. So we came to the conclusion that perhaps there were these kind of distinct neuropsychological mechanisms associated with these receptors, and we went a little bit further and we were able to link these up to specific brain areas. So what we found is if we lesioned the amygdala, we could block the effects of venlafaxine. So we think that these biases associated with new learning are very much the mechanism underlying conventional antidepressants, and we think that a primary target for this is the amygdala, actions in the amygdala. In contrast, we don't see any effect with conventional antidepressants in terms of effects on retrieval, but we do see effects with the rapid-acting antidepressant ketamine, and we've replicated that with the 5-HT2A agonist, as well as manipulating activity within the medial prefrontal cortex. So we've got this nice dissociation both in terms of mechanism, one affecting new learning, the other affecting the retrieval, but also in terms of the brain region. Excuse me. My apologies. Hopefully my cough will settle. So one of the potential challenges that we faced was that when we give a drug, and it's being administered in a task such as ours, the reversal, this moving towards 50-50, sorry, this moving towards 50-50 could be due to some sort of specific modulation of affective state-induced biases. But it may also be that the animals have just forgotten how to do the task. And so what we did is we used our reward-induced bias. We used our reward-induced bias as a control method. And so the animals were tested using exactly the same bowl digging protocol, but here we used our ketamine, and then we used the reward-induced bias, and we were able to show this very specific effect. And we were able to show that for both ketamine, scopolamine, and DBS. The DBS was a deep brain stimulation that we did into the medial prefrontal. If you just bear with me a minute, I'm just going to pause, and I will hopefully sort my cough. Okay, hopefully that's a bit better now, and it will last to the end of the presentation. So the importance of these data are that they suggest that there's a specific effect on an affective state-induced bias by these rapid-acting antidepressants. This is not some mechanism that just involves generalized memory impairment. The animal's cognitive function is completely intact in a control assay, but it looks like we can selectively modulate an affective bias circuit, which, if we find that to be the case, is a particularly exciting finding. So the other thing we were interested in, though, was this sustained effect. And so what we didn't know, we were only looking at 1 hour effects. We were only looking at an effect at 1 hour, but in fact, most of the patient data looks at patients at 24 hours. So we wondered, what is happening 24 hours after our treatment? How are these affective biases being modulated? So we used a same protocol, but this time we injected the drug in the home cage, and we waited 24 hours before we tested the animals in the preference test. So we induced a negative affective bias using a pharmacological psychosocial manipulation, and then we give the drug, and then we test them 24 hours later. So this is after the acute effects of the drug have passed, and we were excited to think that we might see this neutralization of the bias sustained. And in fact, that's not what we saw at all. So when we tested the animals, we saw a complete inversion of the bias. So where the animals under the vehicle treatment would show a negative bias, this had then become a positive bias at 24 hours. And we could completely replicate that by doing medial prefrontal infusions. This is slightly different to what we see with scopolamine. So though scopolamine shows a 24-hour effect, we see the sustained effect, and it's still significantly different. It is not showing that inversion. So there was something very exciting about what we're seeing with ketamine, suggesting it had the capacity to change some state and allow some sort of relearning where the bias could be modulated. You have to remember, this is not a flip from a negative valence to positive. These are all a reward memory. It's just that where that reward memory was relatively less due to the negative affective state, it then flips to become relatively more than the control condition. And we then undertook a series of experiments where we tried to understand more what the mechanism might be behind that. And one of the first things we were interested in is, could you block protein synthesis in medial prefrontal cortex, and could you block the development of this inversion? And that's exactly what we find. So we don't see an effect in terms of this acute modulation. So this is not protein synthesis dependent. We didn't think it would be. It's only happening over that kind of 1 hour time period. But when we use anistomycin, which is a drug which can block protein synthesis, and we infuse it just into the medial prefrontal cortex, what we find is we still see this part where we see the neutral, but we lose the inversion. So it suggests there's kind of two components going on to how the rapid acting antidepressant is working. So what I think we're beginning to kind of show, and what's emerging from these data, and our affective bias test and animal studies are helping us to understand, because we can do these kind of more controlled experiments. We think that reward-related learning and memory can be is biased by the acute emotional state. So this can be induced either using psychosocial or pharmacological manipulations. We've done a lot of different studies now, and it's really consistent. We get nice dose-dependent effects in clinically relevant dose ranges and at clinically relevant receptor occupancies, which suggests that conventional antidepressants are working through this neuropsychological mechanism. It's not one thing on its own. It requires both the biological state to be shifted and experience-dependent behaviors. Delayed versus rapid-acting antidepressants exhibit very different neuropsychological effects in the affective bias test. And we've also got a lot of work we've done in the judgment bias task, which you can also, if anyone's interested, I'm more than happy to discuss more. And then we've got the new data where we've shown that rapid-acting antidepressants seem to have this specific effect where they can modulate an affective state-induced bias. So we've kind of proposed a slight, not necessarily a modification of the neuropsychological hypothesis, but sort of an expansion of it. So we think that for conventional antidepressants, the delayed onset is because of this requirement for new learning. So when people are depressed, the brain has over time developed a kind of bank of negatively biased memories. Those negatively biased memories affect our ability to anticipate future positive events, and therefore, as cues in our environment predict those events, we retrieve those memories, but they're negatively biased. If we change that state using an acute antidepressant or cognitive behavioral therapy, we can positively bias the cognitive processes. But then that patient needs to have new experiences. So it's then those then are positively biased through learning and memory, and that takes time to build up and then to kind of reverse the subjective experience of low mood, and hence why it's delayed. Ketamine and potentially the other rapid-acting antidepressants do something quite different. So we think that they're able to modify these old memories. They're not changing memory. They're not amnesic at the doses certainly that we're using. They're not amnesic. But what they seem to be able to do is have some selectivity towards an affective bias circuit. And so ketamine is able to modify the previously learned negatively biased memory. It's able to either attenuate or even perhaps shift to a more positive state, and that's why we see immediate and subjective improvements in mood. And so we've kind of brought this together into a sort of idea around, we've coined the term turning the glass from half empty to half full. So we think that there may be an interaction between biological and psychological mechanisms that could explain both this very rapid, so within an hour, but also sustained effects of ketamine on mood. And it's not just ketamine. We can link this to the pharmacology of other rapid-acting antidepressants. If we look and zoom in on the prefrontal cortex, and particularly we think these LAIR5 pyramidal neurons are particularly important. We know that ketamine and scopolamine can disinhibit, so they can reduce the inhibitory input, so we can raise glutamate levels in this part of the brain. We also know that drugs like the psilocybin, the psychedelics, that they act on 5-HT1A and 2A receptors. And through targeting this system, what we think you get is this shift in excitatory inhibitory balance. And that may be linked to selective pharmacology. So we think that low doses or certain types of drug are able to very selectively target this circuit. So they're taking kind of almost offline the affective bias circuit without having big impacts on other aspects of cognition and the core processes of learning and memory. We think that generates a therapeutic window. And rather than the drug itself having a sustained biological effect, it's this shift in the excitatory inhibitory balance that is then held and sustained within these circuits, which mean that plasticity change means that negatively biased reward memories can be reactivated. And then we have this protein synthesis dependent plasticity, which then shifts the bias from negative to positive. And that positive bias will lead to subjective improvements in mood and sustained subjective improvements in mood. And so the arising hypothesis, there's a number of things I think that are both important mechanistically for us in terms of the potential to identify new targets for drug treatments, drugs that are more selective. Perhaps we can get away from the dissociative effect and focus just on this 24-hour, this sustained effect. But also, more generally from a clinical point of view, we think the delayed onset is linked to the requirement for new learning. And that means that potentially, we've talked a lot in our field about treatment resistance. And in fact, perhaps treatment resistance has more to do with how we use the medications, and perhaps people on conventional antidepressants, it's really important to look at how much they're able to re-engage with rewarding activities, because if they're not involved in any positive behaviours, perhaps the drugs have limited capacity to work. We have seen evidence of emotional blunting with the SSRIs, and that may limit the potential to treat this very important aspect. So whilst it may improve people overall from being negative to neutral, it may actually limit their ability to go into the positive, and that may have some efficacy challenges. Rapid-acting antidepressants, based on certainly what we're doing, are able to modify these previously learnt biased memories. We think it could be through disrupting a circuit, possibly a common mechanism involving modulation of the glutamate signaling and downstream plasticity changes. And that leads to rapid and sustained antidepressant effects. And I'll finish there with acknowledgements and apologies again for the coughing.
Thank you, Dr. Robinson, for such an amazing session. I'm sure the participants have some questions. So now we'll move on to the floor for some question and answer session. For people who are in person, please raise your hand. For people who have joined us online, please unmute yourself only when I call your name out, and please open your video so that we can make this session more interactive. If anyone in person has a question, please raise your hand.
Let me see that. Thank you very much. Dr. Robertson, this was an amazing lecture, and we were glued to what you were saying. You speak very well, despite all this cough and disturbance. We thoroughly enjoyed your lecture, and we feel sorry that you had to go through this trouble. Your data is just amazing, very impressive data. And so I don't know if I introduced who I am. I'm Dr. Athar Enam, friend of Zafar Bashir. So that's me, Athar Enam. So anyway, so your talk was just wonderful talk. And we will put it on our website also so that we can listen to it sometime later. So back to you. You want to ask questions from the audience?
Please introduce yourself before asking the question.
Hello, am I audible? Okay. Dr. Robinson, that was an amazing talk. My name's Anas. I'm a fourth-year medical student here at Dow. And like Dr. Inam said, we were gripped to the screens. It was an amazing presentation, amazing results. I was just wondering if, so you know how you talked about rapidly acting like ketamine, how they convert negative biases towards positive bias during the 24-hour therapeutic window. I was wondering if you could comment on the protein synthesis plasticity and the extent to which that's, because for example, say our formative experiences, traumas in our past could be a cause of MDD. To what degree is that protein synthesis plasticity prevalent so that it could affect even those experiences? How deep into that sort of experience could you tap into? Is it like, you know, could you speak to the extent of that neuroplasticity, maybe?
That's a great question. So we haven't done a huge amount with this. I mean, these are unpublished data, so it's kind of new stuff. So you do that thing where you start off with let's inhibit protein synthesis and see if it's that. So that's what we know, and we know the brain region that we can manipulate, prefrontal cortex. I think what I am speculating, because it's speculative, we haven't really got empirical evidence, but I'm speculating is that there is something happens through our kind of development whereby we develop negative biases. Probably happens to us all to some extent. Children are always much more cheerful than adults. You know, they laugh a lot and, you know, they see the funny side of life much more so than as we get older. Perhaps we get cynical as we get older, but you could see that as an important developmental change where our brains are establishing their bias. And when we live in a very negative world where we've had a lot of negative experiences, it may be that the brain becomes more negatively biased, and that's what we think we see in patients with depression when we measure these affective biases. And I think that what may be happening with these drugs is that they are doing something like a reset. So the plasticity is not in the memory itself. It's not that we're changing the memories. It's more about this affective circuit. And I don't have any evidence yet. I've got to design the experiments to try and ask these questions. But I think as memories are retrieved, the likelihood is it will draw on information stored in lots of parts of the brain. So there'll be emotional information, and there'll be information about context and people and all of those things. The idea we're proposing is that perhaps these drugs modulate that emotional part. So they don't stop people remembering, but they enable people to remember them with a different emotional bias. Interestingly, the most recent data with patients doing the psychedelic type of therapies, that is kind of what they're sort of qualitatively describing. So people describe that they can still retrieve these very traumatic memories, but their emotional response has been changed to them. Now, it's very, as I say, it's very speculative, but hopefully it's we've got some nice clinical ideas coming, we've got some animal models, so that's our plan, is to try to actually get a better handle on that circuit. What we want to do is pull it apart. Can we pull apart affective bias modification, as it were, from the kind of core parts of memory, and show that you can differentially modulate one part of it without changing other parts of it? Hope that answers your question.
Thank you, Dr. Robinson. Such a nice talk. I just wanted to appreciate you and tell you that we are also working on the adipose brain tissue axis. I'm working on the serum leptin levels, and finding out what circulating serum leptin levels have to do, how are they linked with the depressive symptoms or the epileptic symptoms. So you have told us that enrichment, the environmental enrichment has the link with the depression. So we are also working on that, and thank you.
So I just wanted to, I asked Dr. Zeba Haq to talk to about her work on leptin. She's a principal of Dow Medical College over here, and she's been working on that one. So, since Professor Zafar Bashir is very proud of your presentation, so I just wanted to feel, hey, look, we got someone over here too. So that's all it was all about. All right, go ahead. Yes, there's some student over here. Go ahead.
Good afternoon, Dr. Emma. I am Salar. I'm a second-year medical student in AKU. And first of all, an amazing presentation. So my question was related to, so it has two parts. First of all, you described how antidepressive effects of most antidepressants, and you used fluoxetine as one example, is delayed acting as compared to ketamine, which was immediate effect. But then you talked about the biases. So for example, one was able to modulate new memories in a positive bias, whereas other was able to modulate the older memories. Now my question is, in case of ketamine, because its effect is immediate, its therapeutic action from a clinical standpoint would be for slightly different cases than, for somebody who's suffering from major depressive disorders for the last 20 years. And then of course the continuous administration of ketamine would yield very different effects to the continuous administration of fluoxetine, which has been proven clinically and tested. So my first question is, what would you comment on long-term use? And my second question is, have there been any studies which have investigated the use of ketamine in things such as PTSD, post-traumatic stress disorder, where one very negative emotional memory is actually triggering, this whole spectrum of disorders, psychological disorders. So a drug which tends to modulate negative memories might work in those disorders more than a disorder that is lasting for the last 20 years, and you have to take a drug again and again, because for those we might need long-term delayed acting antidepressants that we're using.
Yeah, so they're great questions. So the first thing, I think certainly the use of ketamine, the other rapid acting antidepressants, the aim at the moment has very much been around infrequent use. So They're not given daily. There have been some trials that have used them more regularly, but the aim would be that you use these as a kind of bit of a kickstart in treatment-resistant patients. So these are patients that haven't responded to conventional antidepressants, and that they're given once weekly or twice weekly initially, and then that's reduced over time. But there is abuse liability, and there are concerns about the long-term use of ketamine, definitely. The effects with psilocybin seem to be longer lasting in the clinical trial, so one dose potentially lasting 3 months. So you're getting a very long duration of action from a single treatment. But you're right, they're different from the kind of mild moderate depression, which is the challenge that we still face. And for those, we've got some evidence that the different conventional antidepressants are not all the same. So we're not sure the SSRIs are perhaps as efficacious as some of the other older antidepressants, but they have a better side effect burden, and so it's a balance. But I think there is, I'm not a clinician, so that's not my kind of area of expertise, but as a basic scientist, I think there is a window of opportunity to learn from this to develop some better drugs for both groups of people. Drugs that may be more efficacious at positively biasing new memories without some of the side effect burdens or some of the emotional blunting that we see with the current drugs available. And in terms of rapid acting antidepressants, we have some very preliminary data that suggests that you may be able to get the 24-hour effect without having the acute dissociative effect. And so there's the possibility that we could have drugs that would be more widely used in patients with depression, that maybe they can help to adjust that circuit level dysfunction. But I think what the neuropsychological hypothesis is very strongly suggesting to us is that it is not just about the biological mechanisms, it's about the environmental factors that drive and sustain depression. And I think we probably need to work more to understand what it is in individuals' lives that are driving those negative affective states, and try to find ways through associated therapies, be that drug-assisted psychotherapies or looking at how you facilitate positive re-engagement in social activities and these sorts of things. I think those things are quite important in how these drugs work, and I hope that as we hopefully strengthen the evidence for a neuropsychological basis for the cause of depression and its treatment, we might see that happening a bit more. I mean, drugs like treatments of PTSD, it's not kind of my area of expertise, but certainly people have looked at it. Certainly the psychedelics are being looked at very much in terms of PTSD, and also particularly MDMA is a particular interest for that.
Thank you, Dr. Amma, and get well soon.
Thank you.
So we have one last question in this Q&A session. Mr. Mohammad Asrar Yusuf has joined us online, and he wants to ask a question. Can you please unmute yourself and ask the question? Mr. Mohammad Asrar Yusuf.
Thank you very much for giving me the chance. Thank you very much. Ima, my question is relating to my further studies. I am a physiotherapist and I have interest in neurosciences. I want to pursue my PhD in neurosciences. Is there a possibility to get me admission in PhD neuroscience in any advanced country? It can cause me a hurdle because I'm a physiotherapist. I have research in Web of Science Index Journal, but I have no experimental researches published yet now. So please give me some knowledge about that. Can I get admission in PhD neurosciences?
So I think it is more difficult to do that, certainly in my field, where, you know, a lot of the expertise we're looking at is people who've got a strong background in, you know, lab-based techniques. And of course, you know, it's very competitive getting into those PhD places. But, you know, in terms of psychiatry and psychiatry research, there is a big neuroscience interest, and it goes much beyond the sort of lab work that I do. And I think there, you know, we work with collaborators looking at, you know, the other factors that are so important in treating mood disorders and psychiatric disorders. And certainly in the UK and in Europe, there's a lot of research into how you facilitate those kind of more positive behaviours, not just as a psychologist through cognitive behavioural therapy, but something I'd certainly look at is a therapy called behavioural activation, and that's much more focused on behavioural modifications and the sort of thing that I would have thought as a physiotherapist might be very relevant. I'm not a particular expert, but I'd certainly have a look at that area. And we've been interested in the relationship between the drugs and behavioural activation therapies, partly because I think it may be a more economical approach to treating some of the certainly in the UK we've got a very high number of patients needing support, and we don't have the psychotherapists and the resources to put them all through that method, but something that is more around behavioural activation and that support perhaps that links into physiotherapy would be interesting.
So that was very impressive. The question asking about something personal in a public forum, and it was further impressive that you answered it with same passion as you give the lecture. So, so thank you very much. That brings to an end of this wonderful Neuroscience conference. This was the last session. We are very grateful to you, Professor Robinson and Professor Bashir. And tomorrow we will have another day. So thank you very much, everyone. And make sure that you get tea. Thank you. We can't serve tea to you, Zafar. We can't serve tea to you, but you know, maybe you're in Bristol together. You guys can have tea together while we have tea over here. All right. Okay. Bye now. Khuda Hafiz.
I think I'll try and find some cough syrup for Emma.
Yeah.
Thank you, everybody.
I would like to call Dr. Sonia on stage to present the certificates. Dr. Sumbul Shamim. Dr. Zeba Haq. Dr. Sonia is on video. Dr. Sonia is on video.
Yes, yeah, Dr. Sonia is present. This is Dr. Athar now.
Lastly, I would like to ask Dr. Sonia to present a certificate to Dr. Athar Inam. मुझे नहीं बोलना। With that, we have ended the sessions for today.
Requested to kindly proceed for the tea.
Thank you everyone for joining us today, and we will resume from tomorrow in the same venue. All faculty members are requested to proceed for tea, and thank you everyone. Hope you enjoyed yourself and learned a lot. Take care. Allah Hafiz.